For us, fruit means a mango, papaya, apple, or a banana. However, for a botanist, fleshy and juicy mangoes or papayas are as much a fruit as are groundnuts or cashew nuts, which are dry in nature. Fruit, the final product of sexual reproduction, develops from an ovary post-fertilization. Incidentally, in most cases, fertilization and the development of fruit triggers the withering of other parts of the flower, including the sepals, petals, and stamens. However, in some cases like apple, floral parts such as the thalamus become a part of the fruit. Such fruit in which other parts of the flower besides the ovary are involved in the formation of the fruit are called false fruit. On the other hand, fruit like mango and papaya that develop only from the ovary are called true fruit. Interestingly, while mango and papaya develop after fertilization, fruit like banana develop without fertilization and are called parthenocarpic fruit. Moreover, in the case of banana, the edible parts develop from the inner tissues of the ovary, and the brown specks that we see are the remnants of the ovules. If you cut open a fruit, you will see that it is made up of two parts, the pericarp and one or more seeds. The pericarp is the fruit wall that develops from the walls of the ovary. The seed, the second part of the fruit, is actually a fertilized ovule. Typically, a seed is enclosed in a tough outer protective covering called the seed coat, which is actually the hardened integument of the ovule. The micropyle of the ovule persists in the seed as a small pore through which water and oxygen enter when the seed germinates. Inside the seed coat lie one or more cotyledons as well as the endosperm, embryo, and embryonal axis. The cotyledons, which are often fleshy and swollen, contain abundant food reserves. Endosperm, on the other hand, is a nutritive tissue nourishing the developing embryo. Interestingly, in certain plants such as bean and pea, the developing embryo completely consumes the endosperm. Such seeds that contain no endosperm are called non-albuminous seeds. However, in wheat and maize, a residual endosperm is seen as it is not completely consumed by the embryo during the developmental stage. Such seeds are called albuminous seeds. Sometimes, in addition to the endosperm, seeds also contain remnants of the nucellus called the perisperm that also stores food material. Such seeds are called perispermic seeds. Black pepper is a common example of a perispermic seed. Whether a perispermic or albuminous seed, as the embryo begins to develop, the seed starts to mature and lose its water content to become relatively dry. A mature seed, therefore, has only 15 to 20% of its mass as moisture content. Moreover, the embryo's growth rate slows, and under unfavorable conditions, it may enter a state of inactivity called dormancy, during which the embryo develops an increased resistance to desiccation. Conversely, conducive conditions that provide favorable temperature and appropriate water awaken the dormant embryo, and the seed germinates, with the embryo developing into a seedling. This ability of the seed to germinate is called seed viability, and it varies across seeds of different species. The seeds of spinach, for instance, remain viable only for a year or so, whereas muskmelon seeds remain viable for 5 years. Did you know that excavations around the Arctic tundra region have led to the finding of seeds of Lupinus arcticus, which remained viable for 10,000 years before germinating and flowering? Seeds, a character unique to angiosperms, also offer many advantages to these groups of plants. Seeds have special mechanisms such as bursting or wings that help them disperse to new habitats, thereby allowing plants to claim new areas to propagate. In addition, the seed's tough outer coat protects the embryo from hostile environmental conditions. Upon germination, seeds also have a better survival rate, as they are guaranteed nourishment by the cotyledons. As seeds are products of sexual reproduction, they help bring about genetic variations within plant species. Genetic variations are the key to evolution and also help plant species adapt and survive in different environments. Seeds, which help in the propagation of angiosperms, also form the crux of agriculture. Interestingly, farmers in recent times have started using hybrid seeds developed through artificial cross-pollination techniques. These seeds guarantee increased productivity and superior crop quality. However, when seeds born by hybrid crops are sown, the seedlings do not exhibit the hybrid characters due to the segregation of characters. This forces farmers to purchase new hybrid seeds year after year, which is an expensive affair. To address this problem, botanists are trying to understand the genetics of apomixis and transfer apomictic genes into hybrid plants. Apomixis is a form of asexual reproduction in which the seeds or embryos are formed without fertilization. In fact, seeds or embryos either develop from an unfertilized egg which did not undergo meiosis, or from the cells of the integument or nucellus. Botanists believe that converting hybrid crops into apomicts will prevent the segregation of characters in seeds produced by hybrid crops, and thereby eliminate the need to purchase hybrid seeds every year, as the seedlings produced by the apomicts will resemble their parents genetically. Did you know that plants such as grasses and those belonging to the family Asteraceae and Rutaceae are common examples of apomicts? Moreover, in certain apomicts such as Citrus jambiri, which belongs to the Rutaceae family, the nucellar cells surrounding the embryo sac divide and protrude into the embryo sac. Later, these cells develop into embryos, and hence a single seed contains more than one embryo. This phenomenon of having more than one embryo in a seed is called polyembryony. Thus, though we have gained a lot of knowledge about the formation of seeds and fruit, there are many mysteries that scientists are still trying to unlock.